mean, it's already, short, probably years. There's some.
All right, everyone, I think we are going to get started. We have Dr. Lorena Endara today. I'm going to let one of her students introduce her.
Hi, so my name is Eden. I've met some of you before. I'm a wildlife and fisheries biology undergrad. And I'm also the herbarium assistant. But here today we have Dr. Endara. Dr. Endara currently serves as the curator of the Clemson Herbarium and as a lecturer in the Department of Biosci. She has devoted her career to plants and has been deeply inspired by high floristic diversity in her home country of Ecuador. She conducted her undergraduate studies in Ecuador and then completed her PhD at the University of Florida, where she studied several groups of chlorophyllid orchids using morphological and molecular approaches. She has a diverse array of scientific interests, and her ultimate goal is to facilitate science and highlight the important role of plants in our society.
Thank you very much, Ida. Good morning, everyone, and thank you for the invitation and the introduction. Today is Friday, so I hope that I will make this fun, that I will take you back to Ecuador for those of you that don't know about it, and also that you see me as a facilitator of science, as Ida just mentioned. So I am going to start with this image for one reason. This image can be taken from the tip of Sassafras Mountain, right? Or you can go to Highlands and stop in the Outlook and it will look like this. And this is what I feel at home here, because montane forests are the part of my research and my interest. This picture was actually taken in northwestern Ecuador in a reserve that is called Siempre Verde. And this is why I feel here happy. After 20 years of being in very flat Florida. Don't take me wrong, I love Florida, but this is my thing. Okay, so who am I? I have been highly influenced by the place that I was raised. As Eden just mentioned, I am Ecuadorian. Ecuador, for those of you that don't know, it's in the equator line in South America between Colombia and Peru. So it doesn't get more tropical than that. So I have always been fascinated by living organisms, and Ecuador is a great place to be, as much as this part of the state is. Here, this is the diversity of Ecuador in numbers. I know that probably not a lot of you might be interested in plants, but I know that many of you might be interested in birds and mammals and so forth. So there are a couple of things I would like you to see. First, the diversity, and involve the endemism. Right? So Ecuador, that is the size of Colorado, and South Carolina is 1/3 of the size of Ecuador. But the diversity index in this country is super high. So it's one of the most diverse countries in the world. And of course, I come from a family that is super into plants, and I have always been moved by plants. So here there are some numbers that I would like you to see. Again, don't forget, this is the size of Colorado. We have the last checklist that is official had 16,000 plants, vascular plants. Now we're closer to 20,000, but we expect to have around 25,000 species of plants, of vascular plants. But one thing I want you to see is the endemism. Endemism is high no matter which group of living organisms you are studying. So Ecuador is an unbelievable lab for us that are interested in life sciences. So the really most important question that I address in my research and in general, it's why are there so many species? And I think that moves us all, right? It moves us from a point of conservation, exploration, what is creating this equilibrium that allows different species to be living in the same place. And that's, since I was probably 18 years old, that has been the heart of who I am as a researcher. So I am showing you, this is the path of my career. I am a person that loves diversity and loves to document diversity, and I like the mechanism by which we do that. And I want to show you a couple of things. The best part that we all know is to be out in the field looking at plants, right? So I do exploration. This is my husband. Before we were married, I took him there. He's a real trooper, and so he has helped me throughout all these years. But again, going back to diversity, I like to document, and all of this works in a very organized fashion. I collect plants that are of my biological interest, but I also produce documents that translate the diversity that I am seeing. So I have been part of monographs, taxonomic and systematic monographs. I have been part of checklists, assembling checklists. So the couple of vascular plants of Ecuador, also the red list of endemic orchids of Ecuador was part of 1/3 of the book for the plants of Ecuador. I do applied research, so don't feel that these are disconnected. Every single thing that I do is really dictating what happens with the species, what are we calling species and so forth. So I do use all the information that I collect in applied research. And this is something that I have been more involved after many years. I have been doing this for probably 20 years. Lately, I have seen the fruits of a lot of this knowledge go into policy, right? So the power that I have gained through documenting diversity and understanding diversity at a regional level has helped me with policy. I am not going to talk about anything regarding to policy because that's not the meat of who I am, but know that there are some really cool things happening in legislation regarding to the rights of nature, for example, that documenting all this diversity has empowered me to do. Okay, so now let's go into the talk, and I'm going to introduce to my talk from tiny orchids to big trees. And this is a picture of some of the systems that I work with. This picture is dear to me because all these cloud and water contribution comes from the Amazon. So Ecuador is cut in the middle by the Andes. So towards the west of Ecuador, we have the coast that faces the Pacific. We have islands in Ecuador. The Galapagos Islands belong to Ecuador. Then the coast, then we have the Andes, the newest formation of the Andes is in Ecuador. And then to the east, there is the Amazon, right? So this is a contribution of clouds from the Amazon. The first picture I show you is the contribution from the clouds that come from the west, from the Pacific. So today I'm going to talk to you about Scapocephalum orchid system, big tree research, and advancing the Clemson herbarium. As Ethan mentioned, I am the curator and I am here to facilitate science. So let's start off white orchids. Do you remember the amount of plants that I mentioned Ecuador has? So from those almost 17,000 species, 1 out of 5 is an orchid, which is astounding, right? And this happens not only in Ecuador, it happens in all South America, right? Orchids are the drivers of diversity in the flores. So in Ecuador, there are like now close to 4,500 orchid species. And the crazy thing is that 40% of them are endemic, right? So they occur only in Ecuador. Think about how incredible this is. It's, again, a lab for evolution, but also a big responsibility for Ecuadorians to protect this. I work with a subtribe, the Pleurotalidina, that is the most diverse subtribe in orchids. And just to give you a global look, this is a phylogenetic reconstruction of the orchids of the family, and the Pleurotalidinae are up here. So how you need to interpret this phylogenetic tree is this is a diversogram. So each of these triangles, the size of it is informing you how diverse that group is. And in the heat map, the colors in this red, really red, bright red, it's informing you that these are really young clades, right? So the pleuropalids are neotropical, and they are the most diverse group. And the only comparison that we have of the diversity is the dendrobene, that is the ecological equivalent of pleuropalids, but in the old tropics. So again, pleuropalids are driving the diversity in the tropics. This is to take it, and again, don't forget, there are going to be a lot of very beautiful pictures, because it's Friday, but also because these groups are fantastic, right? Check out the diversity of the floral diversity of Pterophytes. They are really great, and they're not your typical orchid, right? I am very, very glad that people like orchids, because they're charismatic and they are beautiful, but those are not the orchids that I like. I like the interesting orchids. And I'm going to introduce you to my very first study system. These are Dracula orchids. And when I was an undergrad, I studied this group. So they are called Draculas because they are epiphytic. They grow on top of branches, and the flowers are hanging from the branch, and they look like little bats. So they are fantastic. The reason why this is not part of my research now, but I started like this, and this is a life changer for me because in this group, I found the pollinators, but this opened the door for my education here in the United States because most of the people that work with these orchids are based here in the US. But let me tell you a little about the system. Dracula orchids are fantastic, for not just because they're crazy odd, but also because they produce fragrances that are similar to the fragrances produced by mushrooms or fungi. If you are walking in the field, in the cloud forest, and you smell a mushroom or like a fungal, like the mushrooms here in the supermarket, and you look up, you most likely will find one of these orchids. So what I discovered after hours of standing in front of these flowers is that Zygotica flies that are related to Drosophila flies, the flies that are used for genetic studies, and also the ones that if you leave your bananas go bad are flying on top of them. Zygotica is a genus related to that, but it's slightly different in one thing. They depend on fungi to complete their life cycles. Right? And so they are the pollinators of these orchids, and these orchids have one, as hopefully you have all taken a taxonomy here or in other institution, but one in orchids, one of the petals is highly modified, and it acts like a landing platform for the pollinators. And in this orchid, the landing platform, or this labellum or lip or modified petal, resembles an inverted cap of a mushroom, right? So these are fantastic creatures. Now, what is humbling about this is that I went very innocently thinking that I was going to discover a lot of cool things, and I did. But what was humbling is the number of new fly species that I found, right? Which complicates the publication system because I work with morphospecies for most of the flies that I collect because many of these flies are new to science. And it takes a while to describe them and so forth. But this, again, is what changed my life as a professional, opened the doors, and that's the reason why I strongly believe in undergraduate research experiences, and I try to support them as much as I can. So, as I was telling you, these flies are, and flies in general as pollinators are really slow. And I hang out in front of Dracula orchids for really long periods of time in these systems. This is again one of the reserves that I work in northwestern Ecuador in the foot slopes. And so when I was looking at the flies that were pollinating Dracula orchids, I had a lot of really boring time. So I would walk around and I would look at other systems. One thing that is fantastic in Draculas is that you can find a lot of natural hybrids, but also these Draculas are sought after for horticultural purposes. So whenever you remove them from the natural habitats and grow them ex situ, they will also form a lot of hybrids. So as I was doing my observations in the forests, I discovered a group of orchids, not part of Perothalid orchids, but not closely related to Draculas, that coexisted with Draculas, but have really clean specific circumscriptions. So I could not find hybrids between the system, between these orchids. And check this out, what is fantastic is that you have in one tree 4 species of orchids grown in close proximity. So it doesn't get more sympatric than this, right? So I was wondering, why are these guys not, what is driving this? Why are they not hybridizing like Dracula orchids do? So this turned out to be my next project. And that's part of my doctoral project and what I have carried over from research. And this is one of the things that it's very appealing to me. And I thought that I was going to have the lottery of science and get a Nobel Prize. So in the Scaphocephalums are restricted to the Andes and to the most famous systems also in Central America. You cannot find a Scaphocephalum for kilometers. And then you reach one locality and there are many Scaphocephalums living in the same place. So they really live, whenever you find them, you find many of them and you find several species that live in close sympatry. So it was not just where I was doing my orchid research, this happened over and over throughout the distribution range of Scaphocephalum. So why I thought that I had won the lottery was because, and again, I was young and very innocent. I thought that this was going to be the perfect example of sympatric speciation, right? And as we know, cases of sympatric speciation are very, very few. So I was like, I am going to show the world. And don't forget that I am also coming from this place that we know that these orchids diverge very recently. Do you remember the diversogram? One thing I didn't point out there is that the whole parothalid lineage is 18 million years old. And Scaphocephalum and Dracula are around 2 million year old. So this is crazy, right? So I thought like rapid evolution is happening and I'm going to have like the case of like Darwin's finches. They're going to be like, that's the type of system I was hoping. So Scaphocephalum, Scaphocephalum has 49 species. This primarily in the cloud forest and it's primarily epiphytic. And sometimes you have facultative terrestrial and lithophilic orchids. And again, nice pictures to convey one thing. It's not only the diversity driven by the species number, it's also the phenotypic diversity that I see in these groups. So as I was telling you, they might not be the prettiest or charismatic orchids from them all, but they are super interesting. And here you can see the diversity of the genus. And I'm going to like give you an introduction to Scaphocephalum just because sometimes it's interesting to see this variation, right? In Scaphocephalum, the floral display is primarily driven by the sepals, right? So here we have 2 sepals that are fused and are forming a synsepal. And this is what gives the name to the genus. The genus name is Scaphos, that in Greek means boat-like. So these sepals form these boat-like structures, and they are crowned by these sepal tails, and very reduced here, but these are sepal tails that have function in attracting flies. Flies are attracted to these like hanging structures. So in the sepals, there are like these cushion-like structures that are called osmophores, and osmophores are secreting proteases. And another thing that it's interesting to see here is that this dorsal sepal, and don't forget these are monopods, so the parts come in numbers of 3, so we have 2 sepals and 1 dorsal sepal here. Sometimes they have these cushions might be present in this dorsal sepal here. And again, secreting different types of fragrances. And I am going to now go in, and also one thing I need you to do now is look at your pinky nail and also imagine the flowers I am showing you today are the size of your pinky nail or smaller, right? So everything that you're going to see here is kind of like, and also think about the flies that pollinate them. They are super tiny, Okay, so here you have, I have taken out some of the parts, and so here we have the osmophore. If you take this out, you have the lip that is that very specialized petal. You have two petals that are flanking a column, and for you that need a review, orchids have a structure that is called the column, where the stamen and the carpels fuse, are fused. This is the visuals of the column, and the column again is stamen and stigmatic tissue that has been fused, and this is the tip of the column. And here you can see this little thing here is the cap of the anther. Again, another thing that is important in orchids is that orchids don't have pollen grains that are loose. They have all the pollen grains are compacted in a mass, and these are called pollinia. So here we have the anther cap, and if you remove the anther cap, you have the pollinia, and the pollinia is what gets attached to the pollinator, and this is one of the pollinators of this group. I am not going to talk about the pollination today of Staphylococcus, but I do have that on this. The pollinators are some Saprophyta flies, again, a group that most of the species that I collected are new to science. But check where the pollinia is getting attached, right? So these flies are ridiculous on how much weight they can withstand in their backs and continue to fly. So with this background on Staphylococcus, what I wanted to understand is if the species that occur in each of these places are each other closest relatives, because that will be an indication of sympathetic speciation. So for that, I reconstructed the phylogenetic tree or the evolutionary history of the genus, and I integrated molecular and morphological data. And again, it's right there, and I'm going to take you to Ecuador and let you know that most of the time that I spend looking for these orchids has been the product of many years and a lot of people that support me. And it's probably some of the most enriching experiences of my life for many reasons, right? I think that we all share that whenever we go to the field. So, and plus, I spent all my NSF grant here, so I have to show it off, right? So, okay, this is northwestern Ecuador. My collaborators, some of these places are super remote, and when I went into this field trip, it was where, like, I don't remember when, but I remember I left Quito, that is the capital, to go to this place, and it took me longer to get to this place than it took to a ship to get to the space station, right? So it's kind of odd that this happens in our world, right? But anyway, I reached this remote forest and look for plants, and sometimes plants collaborate better than others. This is in southern Ecuador. There are a couple of species there, but this is in the border with Peru, and there was a war, so there are a limited amount of places that I can go without the threat of mines. But this is a very nice group of collaborators that I have in southern Ecuador, and my very first student, undergrad student, was here. Okay, this is central Ecuador, and for those of you that don't know, this part of the Andes has tons of volcanoes. And here you can see this is an active volcano in Eastern Andes, and here there are like the species that I collected there, and you can see that this little guy was affected by the ash plume that this volcano left on where it lives. So, you know, we have a lot of adventures. This is Luis. I am an Ecuadorian that has moved to the United States. He's my counterpart. He is the American that went to Ecuador and lives there. And you probably have heard a lot because he's super into a diversity index there. So Luis and the Yost statistic is kind of well known. So, and he's a trooper that for conservation. And this is the eastern slope of the Andes. Here I got lost. My husband got lost. My guides got lost. This took 3 years for me to find this species. And here I have to, of course, document how much we look for it. but we finally found it. So again, life is a lot better when you're really good. So you have to go back to the lab and do the things that you need to do, right? So this is a no fashion approach to reconstructing trees. In this one, this tree that you're seeing here has all the Scaphocephalum species. It was reconstructed using five loci. And this is the product of all those crazy times in the field and many hours in the lab. Take home message of this. One is that one of the species, the one that I was looking the most, is not part of the genus. Right? So Scaphocephalum medini is not part of the genus. It's good because it's one publication, right? The other one, Scaphocephalum pleurotelodes, had never been easy to place. And this is the troublemaker of my phylogeny, even though it shows that it's within the group of, within the genus, this species keeps on flipping and losing, making the resolution of the tree lower. So I have always been wondering if this is a putative hybrid in the genus. One thing that is super interesting is that in orchids, hybrids don't occur only among species within the same genus, but also they occur among species of different genera. So they're intergeneric hybrids. So I have been always like wondering if this is the product of an ancient intergeneric hybridization. So anyway, this is how my data was limited by the methods. We're going to talk a little more about that in a second. But one thing I want you to see here is I did not win the lottery. I did get a lot of fun time in the field, but when I came back and tried to answer my question, no, the species that are living in the same ecosystems and living close sympatry are not each other's closest relatives. So I did not have a case of for a system of sympatric speciation. It's more likely that I have a case of allopathic speciation with secondary contact, and most likely what plays a big role here is ecological filtering. But I was telling you that I was also going to gain a lot of, that I was going to use morphological data, and I spent a lot of time getting morphological information and creating this. It's very time consuming, but it's worth it for one reason. Don't forget, we, first of all, are observers, and observations are what makes you think about things differently. Like careful observation makes you think about systems differently. So this is all the process, and I'll be super happy if you ever need to do morphological work to discuss it, because this is probably one of my fortes. But I love extracting morphological characters and assembling in order to analyze them in a phylogenetic framework. Just for you, I don't know how often you see this type of work done, but a person like me that is a systematist uses all these morphology and phylogenetic frameworks to guide the our treatments for taxa, right? Let's put it in other words. I write monographic treatments, or I can write circumscriptions for species that are guided by both phylogenies or molecular work and also morphology. So in this graph, you're seeing the molecular backbone or the framework, and I have traced every single morphological character for the genus. And what I am getting ready to do here is to describe a genus based on this molecular structure or framework using all the traits that are supporting these traits, right? So this is how I right now, or more modern taxonomic treatments are written. So don't forget, I discovered I need to get this guy out, but I am always wondering, why is this putative hybrid doing here? So if you see it from the point of morphology, if I include this guy in Scaphocephalum, I only have 4 traits that describe the genus, right? Because Scaphocephalum is putting a lot of, getting me a lot of noise. If I remove it, I have 12 specimens or 12 traits that describe the genes, right? So anyway, don't forget also that I was telling you that like this putative genus is decreasing the resolution of my phylogeny and it's changing places. I will come back to that in a second. But going back to why is morphology and morphological data important, it's because it helps us think and see things differently. This is one of the examples of many examples that I have throughout the phylogeny that morphological data was super important. And the combination of both. This plate, this is the phylogenetic or the evolutionary history of Scapocephalum, and this is the little plate I am extracting and plotting all the traits that I am looking at. So this plate has the very first lineage that splits is Scapocephalum ophidium that is pollinated by flies. And the rest of them, we don't know what makes them. But here we are seeing a jump, a character evolution, a character jump towards something that might be a different pollination syndrome. And when you look closely, and don't forget that these are really tiny orchids, right? So I have been spending lots of time in front of the electron microscopy, scanning electron microscope, and I have discovered that something that I can tie with the behavior of the fly whenever I see them land. The flies land on the upper part where the osmophores are. The osmophores are super rich in textures. So here you can see an example of how the textures of these osmophores look, right? This ornamentation is lost in this group, right? So I was like, I'm going to collect information for all the group. And so that's what I did. And I found some unbelievable things. First of all, the texture, and it's very hard to see naked eye, but you can see that there is slightly differences, but check these out. And I am just including some of these guys, but I have for other species. But you can see the textures of the osmophores and how they differ. And if you look inside, and again, that's the miracle of like being like, or the great thing of observing carefully, is that there are like all these crystalline formations that are proper for each or specific for each of the species. So crystals, these crystal formations that were unknown to science and to orchids before, came out in every single Scapocephalum species. So I'm going to show you some of these beautiful depictions of crystals here. Before, let me go over, we have like these crystalline formations in cubes, in rectangles, and then we have like these little diamond square things. We have bruises, and we have sand. So as you can see, every time you go into the scanning electron microscope, it's almost like opening like it, right? It's like a jewel. And so there are a couple of things. Crystals in most of the plants are packed in cells. Right? These crystals are epithelial. So we were trying to understand what is the chemistry behind these crystals. And it seems that they are waxes that crystallize, and the function of that is to divert or to reflect the light for flies. Flies are super attracted to glistening surfaces, and it seems that that's the function of these crystals on the surfaces of flowers. And of course, here you can see different densities of crystals. So as you travel through this landscape, you will see places that have more crystals, and these are more dense, closer to where the pollen or the pollinia load is. So they're really guiding the fly in the surface of the flower. So this might be one of the things that attracts Scapocephalum to the flowers. I'm sorry, Sapphirofita to the Scapocephalum flowers. So here we have several flies, so you can see. And that brings me to what I have ongoing projects. I am not a research faculty in the university. I am a service faculty, but I do keep my collaborations with with different friends and great collaborators. And let me start by one of the collaborations. So I was telling you that observation and exploratory observation feeds a lot into your science, right? Here we have one of those scanning images electron scanning images, I started seeing, and this is congruent with the behavior of the fly on the flower. Flies are all the time flapping and they are combing and they are always interacting with these surfaces with the proboscis, right? So the proboscis of these flies are like a sponge. So they're always lapping around the surface. And in a close examination, there were tons of fungi and bacteria growing in these landscapes. And I was looking in the literature, and so these flies, if they get dissected, you will find spores of yeasts in the tract. So some people think that they might be feeding in these yeasts or the fungi that are present on the landscape of the flower. But we don't know for sure. Another thing that that might be happening is that either yeast or fungi are changing or fermenting things on the surface of the flowers, and that's what flies are attracted to. In any case, one of the things that we are doing is microbiome of the flower surfaces, and what we have been doing so far is things that grow in greenhouses. So I have all the collections that I show you are now living in Atlanta Botanical Garden. So I have them there. They're in the same space. So if you go there, check the section where they have the Guyana Plateau. They have all the plants from the Guyana Plateau. Just next to the Guyana Plateau, there are all my orchids. So I have them growing in the same very small section, so they all have the same conditions and are subject to the same type of colonization of fungi. And what we have been trying to do is to run microbiome assays for all of these flowers. And you can see I have buds and I have flowers, open flowers in the same inflorescence, and they are all growing in the same place in Atlanta. So what we are finding so far, we have analyzed microbiomes using ITS. What we are finding so far is that colonies of fungi are present in all of the orchids that I have been growing in Atlanta. But there is filtering, and the communities are very different from species to species. Basically saying that the idea is that they're the volatiles of these plants are probably filtering the communities of fungi, bacteria, and so forth. So now you're like, oh, but what about big trees? Of course, I build evolutionary trees, and that's the big tree. So I told you that my past tree was built with 6 regions. So I don't know if you saw the number of base pairs. There were just only 6,000 base pairs. So what you're going to see here is with my postdoc and now collaborator, Gordon Burley at the University of Florida. We have been able to reconstruct using genomic approaches and using around 500,000 base pairs. A new tree for Scaphocephalum. We have a very nice resolution, very clean, and it's supporting that Scaphocephalum medini. Do you remember the hard one to get? It's out of Scaphocephalum, and also Scaphocephalum pleurotelodes keeps on the same position. And now I just finished doing this analysis like 2 days ago. What we're going to be looking at is because we have more information, more markers, and these are all nuclear genes, we're checking what is going on if we can get some type of signal for hybridization and understand if it's a intergeneric or interspecific hybridization. Okay, so for those of you that are more like into big trees, not just big phylogenetic trees, but big trees, part of my postdoc, I changed from all this floral focused research to working with conifers and gymnosperms. And I am not so much into the exploration. I have been more developing probes that will allow us to do genomic work. And I have also been collecting in botanical gardens and creating like a capsule collection for gymnosperms that will be served for reference. But we have samples so far, most of the gymnosperms. And here you can just see some of these numbers and also all of the botanical gardens where we have collected. And I want to a couple of things, this, new sequencing approach, that is a genomic approach, it's a designed approach, we have 405 nuclear genes. This is a completely different power that this approach is giving us. And we do have most of our reconstructions are done with conserved exons, but we do have banking regions to resolve interspecific or like more closely related relations with strips. Sorry for the redundancy. Okay, so behold, this is the most comprehensive tree of gymnosperms that includes most of the species of gymnosperms in the world. This is going to be turned into probably one of those textbook figures. But we are on the working because we still have to like clean a little the data, but this is our final product. And for those of you that are super into plants and evolution, we are supporting the Natalis sister to kinase because normally Natalis appears next to angiosperms, but we support Natalis sister to kinase. I would be very happy to talk about any of these afterwards, but I am going to keep on moving and again going back to trees. Because of this work, I have been super into Araucariaceae. This is the family of the North Pole pine. And these are southern hemisphere heavy families, but they are fascinating and they play a big role in the. I am very connected to the use of plants and in the southern hemisphere. Araucarias were food or serve as food, or the cones serve as food to many of the ethnic groups in southern home. Right, it's kind of equivalent to the pine nuts here in the United States in the Southwest. So I love them. How can you not love them, right? And the power that the process that I just showed you, and also the framework that we built for the gymnosperms, what has given me is this power to understand what is going on at a familial level in Araucariaceae. It has 3 genera, Araucaria, Agathis, and Bulimia. We probably haven't heard much about these 2 other genera. This Agathis is really cool because even though it's a conifer, I'm going to show you in a second a picture, the leaves look like a dipod leaf. And Bulimia is only one species that it lives in Australia, and we don't know where. But it's widely cultivated in ex situ, but the natural population has only been found once. Okay. Everything looks great. Agathis looks nice. Araucaria looks nice. Everything that is in yellow are things that are in New Caledonia, and they're really messy. So we cannot find groups that are clearly defined by molecular data. So what we are thinking is that there is rampant hybridization, intergeneric hybridization in both of these groups. Why is this important and why I'm showing you this is that we are still discovering these really cool evolutionary things, especially in islands in New Caledonia, for example. But Araucariaceae and these trees, they grow where there's rich mines or rich sources of nickel. And so our phones use that for batteries, right? And now that we're changing to technologies, we use a lot of nickel and other minerals that are harvested in Caledonia. So that's one of those pressing things that I always like to bring up because yet we really don't think about this. These Araucariaceae are really, like, you can find an Araucaria growing on top of like a block of one of these minerals. So anyway, this thing that we're looking at a molecular level, it's also being reflected in the classification. This is a herbarium specimen of an Araucaria that we still cannot put in. Here you can see the determinations throughout the years, and we cannot have the certainty of which of the species this one is. So we still have even though we have all the technology and genomics and so on, we still have challenges in many of these fields. So here you can see this is really messy. So each of these little dots is showing you where that species falls in the tree. So as you can see, there is like pretty messy results, but we're working on it and we will figure it out. And very quickly, I'm going to show you a couple of things. I don't know how many of you have been in the herbarium. I am, as I was introduced, and very thankful for that. I am the curator of the Clemson Herbarium, even though my research is not represented in the Clemson Herbarium because my Clemson Herbarium is a primarily regional facility. I do see my role in the Clemson Herbarium as a science facilitator. So for those of you that haven't been in a herbarium, we're a collection of dry plants. How many of you have been in the herbarium? Okay, we need to like change this.
Yeah.
You have to come, you guys have to come and visit me and I will be happy to show you around, but we are a library of plants, of dry plants that have a lot of information. Here we have, let me see if I have a better one. Yes, so we have information here that tells you where the plant was collected, when was it collected, what was living around it. Here we have the plant, and I want you to think about this being also tissue that facilitates DNA studies. The approaches that I show you, the genomic approaches, can work with degraded DNA, so these can be really good resources for DNA. All the specimens that we have are in a database, so you can access from your sofa if you would like to. Here, this is a portal where all of our information is CERNIC. So again, you can go to CERNIC and check the Clemson Herbarium and get any of the specimens that we have. So this is how the herbarium looks, but I hope that you come and visit. We have a certain very systematic way of organizing things, like in libraries, right? We organize using the names that we so hard try to define as I show you. And so we have that. And we are pretty good. We're the second biggest herbarium here in South Carolina and a very good representation of this region. And we have botanical specimens, but also fossils and fruits and seeds and really good literature that you can use. We also, even though fungi are not plants, we are friends with fungi, as I showed you earlier on. And because fungi were parts of plants at some point in history, we have inherited all these things. We have this really old, beautiful collection of fungi that survived the fire of 1925. So if you want to come and see it, you're welcome. We have lichens, we have bryophytes, and so we are the repository of the flora of South Carolina and the region. We're part of the Museum of Natural History and the Department of Biological Sciences, and we are located in the basement of one floor. The role of the herbarium as a science facilitator, and also my mission is to, on top of facilitating research, I want to reduce plant blindness at different scales. So it can be like with little kids, it can be with adult education and so forth. So these kids were yesterday in the herbarium and they were the ones that designed the colors and the font of this presentation. So I think that it's a very enriching, both ways, really enriching experience. Here, one of the things that I would like to show is that even though I teach plant taxonomy in senior seminars, one of the things that I do is also serve other classes and other initiatives that want to show the herbarium and plant diversity. So here, this is like plant taxonomy. We went mini golf playing, and it was an eye-opening experience to see the diversity of plants in that place. So anyway, we serve many classes. Here there are other classes. We also, as a mission and as a curator, I like to build skill sets in my students and volunteers. Here I have already put 2 persons in the job market, so I'm very happy for that. And hopefully I will keep on putting many more. I offer sometimes opportunities for work, and there are tons of outreach opportunities. If you ever want or have something, you can like contact me and I will be happy to facilitate those. So I facilitate those at the college level, department, and also at a broad society. So this is to wrap up the conversation that and this talk. My vision here is not very different from the vision that I had at the beginning of the talk. I like to explore, I like to take care of vouchers and facilitate these vouchers for different type of treatments or different type of policy. And in the way, I love to facilitate a better understanding of plants. And with that, I want to thank you and take you back to Ecuador. And this is another system that I didn't talk about. These are the tigre orchids. That is one of those rapid speciation examples in the Andes. And with this, I'm sorry it took a little longer, but I will be happy to answer any of your questions. And they can be related to research or anything about like the herbarium or Ecuador or whatever you would like, or classes and so. I'm going to tell you one thing. I cannot grow anything. So even if it's orchids, I don't grow orchids, I just praise orchids. And I am far from being an expert in the flora of South Carolina, but know that the background that I teach in my classes and also that I brought from my education is buildable, and I am growing in my knowledge of South Carolina, which is incredible because South Carolina, it's one of those, and this part is one of those hotspots, right? That has a lot of endemicity. So I am really happy to see families that I have never seen before. So now questions. Let me go first and then you. Yes.
So you do a lot of genetics and genomics, collections, preserving, and like scanning electron microscope stuff. How did you get into, I'm curious about the SEM stuff, like you've got, is that one of your collaborators? Are you doing all of these things? You like looking through?
I probably, yeah. So I have devoted 20 years of my life to this. So I have been able to secure funding and explore different things. At this point, I cannot do everything. So that's one of the reasons that I love, and I love to have students helping different aspects of it and take different parts of the project. So for example, our area, I love it, but I don't have the time or the resources to go to, or the legs and the stamina to go to New Caledonia. But yeah, so again, I think that there are different ways of approaching science. One of it is like you can have this fixed project that you know exactly what you're going to do, but another one that might take a little longer is to do more of an exploratory, to have an exploratory path, let's say. Your advisor might not like it, but it's your education, your profession, right? And also be integrative. And that's one of the things that I love of American academia, that you are educated to be integrative. And so you can like make interdisciplinary collaborations and research, and that's welcome. So that's one of the great things. And that's what I was telling you. The Dracula opportunity was really an opportunity that opened the doors to a completely different type of making science here in the US. Yeah, and I will be happy to. Clemson has a wonderful facility for scanning electron microscopy and nanoscale stuff, so take advantage of it because that doesn't happen in other universities. Okay.
I was wondering, did you bring the orchids back to the US to do the electron microscopy and phylogenetic stuff?
Yes.
How difficult was it to get
That
these
is
orchids? into the
great.
country?
That is great. So do you remember the policy part? I got super into policy. One of the things that I got was into policy because orchids are all regulated by CITES, the Convention of Trade of Species, right? And I learned a lot about legislation of different countries. Oh, before in my past life, I was an officer for NGO IUCN, and I worked for traffic. So I got a lot of the knowledge of legislation because of that, and I understood how CITES worked. So with that knowledge, I was able to like go to the officers that administrate the convention and get all the permits. So to answer your question, there was a lot of permitting involved and a lot of talking And the moment that I got into the States with 1 year worth of tissues, I got stopped in Miami because someone didn't understand that I have everything in line. I got the 1 week, my whole shipment was in Miami. And I don't know if you know this, but if you get something that is illegal, they will burn. And so it was the worst Christmas ever. I think that, again, that's another angle that you learn. I did bring everything for phylogenetic research with permits, because if you publish without permits, like, they can even, NSF will cut the funding for your institution. So you have to be super responsible. But yeah, it takes a lot of, as you know, also the US requires permits that are really hard to get for some regions. So
I had to recently get genetic samples, blood and skin samples for crocodiles from the Dominican Republic into the country, and it was really, really difficult. Took a couple-
So where was it difficult? In the country of origin or here?
Here.
That makes sense.
What took a couple weeks in Dominican Republic took well over a year here to get done, so.
Yeah, so that's like, and you know, it's interesting because many of the funding agencies don't take that into account. So for example, I spent all my NSF money and I had no job while I was working. Well, I had no work and I was waiting for the permits in Ecuador, right? I have to ask for a leave and all that in Florida, and like my visa was a mess. Because I was waiting for the permits to go through. So yeah, it is really difficult, but it's not impossible. That's one
No,
of those
it
things, you
happens.
know.
Eventually it happens.
Yeah. Any other questions?
Thinking about the crystals that you were finding on the orchids, you mentioned that they are reflecting different wavelengths of light, right? I know Ecuador also has like a high diversity of hummingbirds. I was wondering if any of those pollinate these orchids, and if you think maybe those crystals are reflecting ultraviolet light that some of the pollinators might be attracted to.
That's a great question, and let me go to one of the slides here. Oh, and I didn't check the chat, so 1 second, I will check the chat. And of course, it has to be here. So and this is, thanks for that question, is a beautiful question, and I have thought about this. With this clay, do you remember I mentioned that this clay changes from having the coloration and the morphology of the flower is different, and at one point I thought that I might have hummingbirds pollinating these plants. But because I had also the wealth of morphology, I compared what lives in the forest where these species occur. And I am forgetting, but it's one of the smallest hummingbirds in the region. But the beak is too big for it to accommodate a hummingbird. So the aperture and let me show you, like this aperture or orifice is too small for a hummingbird to be a pollinator. So most likely we have a case of either moth or butterfly pollination. Now going back to the question of refraction, this whole group loses the crystals. So they don't have the ornamentation, and that's what I was mentioning. The ornamentation is only for those orchids that are pollinated by flies. And I think it has to do with how they are seen or how they are locating the flowers. So in the ecosystem or in the environments where these flowers live, it's really dark. So most likely the visuals are first are targeted by the sepalid layers. This ornamentation that each of these flowers has. That's for flies, but for hummingbirds, it's unfortunately unlikely. Can you imagine an orchid that is pollinated by a hummingbird? It's the best, right? But going back, there are some other orchids, not in the Chlorothalidinae, there are Elanthus and Sobralius that are pollinated by hummingbirds, but they don't have this type of crystal information. But going back to the crystals, they are more than reflecting light. I think it's just they are like magnifying light, so that attracts the light. But I wish I would have like, I was just envisioning this super tiny, and that's why collections are so incredible, because I was able to go and measure all the beaks of the hummingbirds in collections. And just try to figure out with Lori's. Let me check the chat, and I am sorry, I abandoned the chat. Oh, no.
I fixed it.
Yeah. So let me make sure that. Oh, okay. So I didn't have any questions, but I did have the class. So. Okay, guys, any other questions? Anyway, if you are too shy to ask, but you still have a question, come to the herbarium and I will be happy to discuss. And if you ever want to like run any ideas, if you want to like figure out a research in morphometrics or in, we have the primers and all the gene probes for phylogenetics. So let me know and I will be happy to guide you through. Okay. Thank you.
Wow.